What's good, y'all? Welcome back to another edition of Mr. Fitzgerald's biology class. Today we're going to wrap up our third unit on cell division by talking about two kind of special case scenarios with non-disjunction, as well as we're going to talk about what a karyotype is, because those are two important vocab words that kind of wrap up our unit and wrap up our knowledge for cell division. So, real quick, I did post a video below this one on Canvas that kind of goes through the steps with animations that are three-dimensional, so that way you can kind of view those and peruse them and kind of get a better understanding of what's happening during meiosis, during both of the divisions. It does help a lot of students kind of visualize the process. Pictures are good, but sometimes these 3D animations are a little bit better, so make sure you check that out as well. All right, so we actually have talked about the differences between mitosis and meiosis. Remember, both of them go through an interphase step. So the top part here is talking about the interphase they go through where DNA is being replicated or copied. That's during their S phase interphase. They're also doing some growth and development of other organelles, so that way when they divide, every piece has everything it needs. But the reason that it kind of darkens up here is it's talking about the amount or concentration of DNA. So the concentration doubles here because we're replicating the DNA in each case, and then it gets divided up into cells. Now, as you can see here, we start with a diploid cell in mitosis, and then we end with a diploid cell here. So that number never changes. So if it's a human cell, like a skin cell or a body cell, it has 46 chromosomes here. Even though the DNA has been duplicated, still 46 chromosomes. It does not increase its number. And then it goes down still with those sister chromatids being broke apart, those duplicated chromosomes being broke apart. Then we end up with two cells that have 46 chromosomes each. So that number stays consistent as a diploid all the way through. On the other side, with the meiosis, we start off with a diploid cell. Okay, this will happen in one of the sexual organs, either the testicle for a male or the ovary for a female. The DNA is duplicated there, so it still gets that 46 chromosomes still here, but just makes sister chromatids or replicated DNA. Then it actually divides these during anaphase 1 to make haploid cells. What it does is it separates those homologous chromosome pairs that pair up during this phase. So now I still have the same amount of concentration of DNA because it's duplicated chromosomes that are attached to each other, those sister chromatids. And then we break those sister chromatids apart, getting a much lighter color down here and resulting in our 4 unique haploid cells. Mitosis makes clones, so they're identical. Meiosis makes haploid cells or gametes, which are unique because they have a mixture of DNA from crossing over in prophase 1, and also how the homologous chromosomes are broken apart kind of dictates their uniqueness. All right, so anytime that a cell has, there's a cell that divides, there's always a chance for a mistake to happen. Anytime you do anything, there's a chance for a mistake. So with meiosis, there is a much greater probability for error because you have multiple divisions. Instead of mitosis, you're just copying the DNA and breaking it apart. You can't have errors with that, but it's less likely to happen than if you're breaking apart DNA in 2 separate rounds through 2 specific divisions. So how could mistakes be made in separating? Well, simply put, maybe we don't separate the homologous chromosomes. Maybe both chromosomes that are homologous end up in one cell instead of the other. So this could result in too much DNA in some of the cells and too little in others at the end of meiosis. Or maybe when we get to those phases where we break the sister chromatids apart in meiosis 2, maybe those chromosomes don't actually break apart, and I get the full X that moves to a side instead of half of the X. So we can end up with too much DNA or too little. So let's talk about that process, and it's called non-disjunction. So non-disjunction is when you have some type of thing where the chromosomes don't split evenly. So within meiosis, this can happen in meiosis 1. So let's say that, metaphase 1 is happening here. So this is like a normal dividing cell at this phase. Okay, this is an abnormal dividing cell, right? They're supposed to line up with the homologous chromosomes side by side, which they do. But when they're being separated and being pulled apart through anaphase 1, maybe 2 of them get stuck together and get pulled to one cell. So it's going to result in more DNA over here than there is on this side here. So you can see that there's an increase in DNA. You've got now 3 chromosomes on this side versus the 1 chromosome over here. And on this side, it did a normal division, which is supposed to be a 2 and 2 in this cycle. Okay, so they're showing, okay, what's the long-term process that can happen if we have the screw up in the beginning? Well, you end up with more DNA on this side, less DNA over here. Now every cell that's made has a mistake to it, okay? Now I have less DNA over here. I have single chromosomes, just one chromosome in each one. And over here I have 3 chromosomes in each one, when they're all supposed to have 2. Okay, so the process is going to result in either too much DNA or too little. When we have too much, right, if we get that extra copy that moves over, it's called a trisomy, right? You get an extra chromosome. Instead of having 2 chromosomes for something, you get a 3rd chromosome for that same thing. Or if it lost a chromosome, it's called monosomy. So it is 1 instead of being 2 for that copy. So both of them can cause issues for the organism if this cell is used during reproduction. All right, so again, it can happen in the second phase too. So maybe the first division works out great, the homologous chromosomes split perfectly in anaphase 1, but in anaphase 2, the X never breaks apart here. So this is the non-disjunction event. And if you notice now, 2 out of the 4 cells are different, okay? They have the trisomy in 1, the monosomy in 1, and then we have 2 regular haploid cells over here. So this would be the desired 2 from the division, and these would be the more abnormal ones, the ones that really are from a mistaken division here. Now, what does that possibly mean for organisms? Well, usually if you have a trisomy or a monosomy, it results in some genetic disorders like Down syndrome is a good example. I do have some that happen if the sexual chromosomes don't split as well. Most of the individuals, if that is, the offspring that's produced from one of those events, they can live full lives, but they definitely have a disadvantage. Either they have too much DNA or too little DNA, and that results in some visible characteristics, maybe some mental characteristics that are pretty apparent for the organism, but luckily with therapies and everything like that, those individuals actually do fairly well in the long term. So let's talk karyotypes. Karyotypes is just like a way of mapping DNA and kind of putting it out there in a chart. So here's a human karyotype showing you kind of the breakdown of the chromosomes. Now they call this one a normal karyotype because it doesn't have a trisomy or a monosomy. Everything is just paired up normally. Okay. So the chromosomes are actually ordered and numbered 1 through 22 based on their size. So if you can see chromosome number 1, it gets smaller and smaller as you go through the chart. As you get down to 22, those are the smallest chromosomes we have. And then on the 23rd pair, we have our sex chromosomes. So we put the male or female characteristic at the end, okay? So in this one over here is a male, and I know that because it says XY. Males have a Y chromosome, which is much smaller than the X chromosome. It holds a lot less information on it. And females have 2 X chromosomes. And that'll come important when we talk about inheritance and genetics, because there are specific traits to the X chromosomes, and it's why males have a higher percentage chance of being bald or colorblind. Because those genes are on the X chromosome, and males only have 1 of those to work with. So there's a higher chance that they might actually have something that is a recessive trait, which we'll talk about in the future. All right, so the autosomes in these karyotypes, and here's one of a Down syndrome karyotype that has a trisomy at the 21st pair there. These are the 22 chromosomes. So basically an autosome is called a non-sex chromosome. So any one of those first 22 pairs is an autosome, right? If there's an individual that has Down syndrome, talking about that non-disjunction event, they'll have either a trisomy or a monosomy at that 21st pair. So that's an easy way to kind of identify if you have a karyotype. And they do some testing for high-risk pregnancies, specifically with older females that are pregnant, to kind of see if this might be a case. So that way the family can be prepared for it and make sure that they have all the information they need. All right. So there are some other sex chromosome characteristics and disorders. So it can happen with non-disjunction in the X chromosomes. So they show you a typical female, double X there, and then the typical male, the XY. And there's a whole bunch of different kind of issues. Like you can have just 1 X chromosome as a female and nothing else, or have an extra X chromosome. You can have an extra X chromosome as a male. You can have an extra Y chromosome. And you can just have no X chromosome. And notice how the X chromosome is essential. The organism will not survive without an X chromosome. So if for some reason a non-disjunction event happens where it's only the Y chromosome that's passed on, the organism is not going to survive. But there are some different disorders that can happen with this one as well. But they're interesting to look up, but they're not something I'm going to test you on, so I'm not going to really go into those at all. So basically that ends our unit on cell division. Make sure that you do the practice problems that are on Canvas. We are not going to have a quiz for this unit because we're going to wrap this unit, unit 3, in together with unit 4 and do a test on both of those since they're kind of short units together. So you will have to be held accountable for this information in the future on a test. But as of right now, make sure you go onto Canvas, do those practice problems, use your notes, make sure you get a perfect score on it because you get multiple attempts, and reach out to me if you have any questions. I hope you all have a great day and see you later.